Assalamualaikum and a very good day to everyone. My name is NURUL IZZATI Binti Othman from UiTM Perlis. I will present my final year project entitled A Review on Antimicrobial Film Packaging from Pomegranate Peel Waste under supervision Dr. Nafiza Ismail. First, let's begin with introduction, which consists of background of study, problem statement, research question, significance of study, objective of study, and scope and limitation of study. Pomegranate peels are generally among the most frequently wasted items as they are usable. Approximately 78% of the peel is recovered as a residue during pomegranate juice processing. Numerous studies have shown that PPE has instant antibacterial action against the wide range of foodborne pathogens, and it also increases the post-harvest shelf life of food. Antimicrobial food packaging is the packaging of food with antimicrobial to protect them from microorganisms or bacteria that could be harmful. One study estimates that over 30% of the world population suffer from a significant health problem due to the foodborne disease caused by consuming pathogen-carrying food. The most recent data from the World Health Organization (WHO) suggests that over 600 million people become unwell and 402,000 lose their life due to the eating contaminated food. During the past several decades, the world reliant on the petroleum-based synthetic polymers, also known as plastic, has grown to be the most popular material for many applications. Plastic waste has been a significant problem for years because of its excessive uses, difficulties in the breakdown, and enormous quantity which severely influence landfills and water contamination. There are 5 research questions. Why pomegranate peel waste is used as antimicrobial in the film food packaging? Which extraction method is the most attractive for the extraction of bioactive compound in pomegranate peel? What factors affect the efficiency of the extraction of pomegranate peel affect antimicrobial activities? How will pomegranate peel be helpful in the field of food preservation? Antimicrobial packaging, a subset of active packaging and controlled release packaging, impregnate antimicrobial into food packaging film material and releases it over time to kill the pathogenic germ harming the food product and prolonging the shelf life. This health potential of pomegranate is known to vary significantly over the varieties, growing conditions, cultivation practice, stages of development, and the extraction method. This research also specifies the objective, which is to highlight the use of pomegranate peel as an antimicrobial food packaging and characterize pomegranate peel as antimicrobial, and to review the characterization, extraction method, biochemical changes, and the food preservation of pomegranate peel. This study, however, only focused on the antimicrobial film packaging for pomegranate peel waste. The use of pomegranate peel as antimicrobial food packaging and characterized pomegranate peel as antimicrobial are discussed. Additionally, the characterization, extraction method, biochemical changes, and food preservation of pomegranate peel are also being reviewed. However, this study will not cover the other potential of pomegranate peel waste as a food packaging. Now, moving to literature review, which consists of 5 main parts, which are properties of pomegranate fruit, biochemical composition of pomegranate peel extract, antimicrobial activities, and biochemical changes in food preservation of pomegranate peel. The remaining solid waste from pomegranates after juice extraction, green and seed, contain various bioactive and nutrient components such as flavonoids, hydroxybutyrenines, and fatty acid. Figure 2 shows that weight percent composition breakdown of pomegranate parts. PPE is high in bioactive compounds, mainly phenolic acid, flavonoid, and hydroxybutyrenines. The primary phenolic acid identified from PPE are allergic acid, gallic acid, caffeine acid, chlorogenic acid, crigic acid, folate acid, vanillic acid, picoumaric acid, and cinnamic acid. PPE are an excellent source of flavonoid. Flavonoid content and composition are also known to vary significantly among varieties and growing conditions. It was also stated that both concentration of petrochemical and antioxidant activity are highly dependent upon the solvent used for the peel extraction. Extraction is a process of separating and receiving a desired substance or a group of substances from a plant's raw material using the solvent-based technique, soft-tip membrane assistant, and instrumental method. It is a process in which a distance is transferred from a solid to the liquid piece. With extraction technique, an insulation of a target substance from a mixer is completed by contacting a solvent that dissolves it selectively. Depending on the raw material, a different extraction technique is applied. The disadvantages of the low efficiency, high processing time, high cost, and environmental consideration of the conventional extraction method, such as simple stirring, lead to the investigation for a new extraction process. So far, several extraction methods such as a pressure-applied extraction, enzymatic extraction, ultrasound-assisted extraction, recently the vacuum microwave aqueous-assisted extraction have been presented for the extraction of the PP. In a study completed by Xin Wang et al. 2011, PP extracted using a magnetic stirrer with a stirring speed of 1200 rpm at 25 degrees Celsius and using 50 ml of water for 60 minutes. In another research study, Hu et al. 2010 showed that water was an efficient green solvent for the extraction of antioxidant from pomegranate mash, achieving high phenolic content and DPPH cavitating activity in 2-minute extraction time. Ranchbard et al. 2016 used as instant control pressure drop process (ICPD) as a statutory pretreatment for the enhancement of the extraction efficiency of phenolic compound from PP. Pressurized water extraction was investigated for the extraction of polyphenol from PP in the study of Cam and EC 2010. Both of the study need 60 minutes of extraction. Hayden et al. 2021 stated that the extraction of ethyl acetate only take 10 minutes, while Akhtar et al. 2015 extract using ethanol and water take 30 minutes using the UAE method. Compared to enzyme-assisted supercritical fluid extraction, Mushrak et al. 2015 stated that 85 minutes time was taken for extraction using this method. Kadaris et al. 2019 analyzed the UAE and MAE extraction of PP in a comparative way and concluded that MAE was a more effective method of extraction that produced 199.4 mg of dry PP after 1 minute of extraction. A previous study by Alexander et al. 2019 emphasized that the higher antimicrobial activity against pathogenic bacteria was noted from 300 MPa. The Ham et al. 2010 investigated the antimicrobial activity of extract derived from different parts of pomegranate was tested on 7 bacteria. Ali et al. 2019 reported that PP inhibited the growth of S. aureus and Salmonella. The ethyl acetate extract of pomegranate peel was previously reported by Machado et al. 2002 to include high concentration of tannin, which are very active against S. aureus strain. Dia et al. 2011 reported that the water extract of pomegranate peel was also to control gram-positive significantly and gram-negative bacteria. Eshobini et al. 2016 noted that the PPE 20 mg per ml exhibited complete inhibition of spore determination and 75.5% inhibition on mycelial growth of fungi. Phenolic profile and antimicrobial activity of GAPC PPE were studied by Karchoffield et al. 2018 inhibit cellular receptor of pathogens. In a recent study, El-Katabi et al. 2020 studied the in vitro and in vivo effects of aqueous PPE on the brown roots. Instant pharmaceutical activity of PPE was then reported against B. cinera, P. digitatum, and P. expansum by Nicosia et al. 2016. 20 hours of incubation with PPE resulted with almost complete inhibition of all fungal spores at lemons and grapefruit. Domenico et al. 2019 investigated the antifungal activity of 21 different pomegranate genotypes. Researchers noted that all of the tested genotypes have varying antifungal activities against F. ostiporum and FSP Lysopelcici, where some of them completely inhibit the fungus and some others at deficient inhibitory activity. Karchoffi et al. 2018 studied the 2 different extracts of pomegranate peel were incorporated with citosan and locust bean gum for controlling P. digitatum and orange fruits. Results suggested that the addition of 0.361 gram water PPE into both locust bean gum and citosan coating significantly reduced the P. digitatum by 28% and 49% respectively due to the high contents of phenolic compound. Most of bioactive compounds which are abundant in the PPE were previously tested as natural additive for improving the preservation quality of food. The PPE was also noted to reduce the water vapor permeability of citosan-based film material and its antimicrobial activity. Another recent study showed that the incorporation of different concentration of PPE with moonbeam protein film provide a biofunctional edible film for packaging of food product. The incorporation of PPE was reported to give the flexibility to the film, increase the thickness and water vapor permeability, and decreases the moisture content. It was determined that PPE retains its semicircular structure in bio-based edible film and improves the efficiency of the material. In a similar study, incorporation of PPE in citosan was tested for fruit packaging. The addition of PPE to active polyvinyl alcohol (PVA) composite film also resulted in high antioxidant activity and antibacterial ability. Next, conclusion and recommendation. In conclusion, it is proven that pomegranate peel can act as a possible antibacterial agent. The extraction efficiency of the compound derived from plant tissue can be affected by various factors, such as the method of extraction used, the time and differences in the mixture of solvent used, and the utilization of multiple materials. The bioactive substances in pomegranate peel can inhibit the growth of different microbial species. Numerous scientific studies demonstrate that pomegranate peel extract is highly efficient against numerous foodborne pathogens and prolong the shelf life of harvested food. To make pomegranate peel more widely used in food preservation, the following aspects need to be further studied and explored. Study on active antimicrobial component of pomegranate peel. Pomegranate peel composition is complicated. Although the chemical composition of pomegranate peel was well studied, the number of specific studies about the mode of action is relatively few. Let's study on the antibacterial mechanism of pomegranate peel. Pomegranate peel has an excellent bacteriostatic effect, but the mechanism of its bacteriostatic effect has not been studied instantly. Furthermore, research on the mechanism of bacteriostatic impact will be helpful to promote the application of pomegranate peel in the field of food preservation. That's all from me. Thank you and have a nice day, everyone.